Good morning, everyone. Let's start the talk. So these are the 20 amino acids which is the base of all the living system, and these are non-proteogenic amino acids which also exist in the system. So have we ever wondered why only these 20 amino acids and why not others? And is it possible to incorporate all these non-cognate amino acids or non-proteogenic amino acids inside the growing polypeptide chain and ultimately into the protein? So is it possible? So people have done that, people have successfully incorporated those, and they have shown that it is advantageous to the system also. What are those advantages? So by specifically incorporating these non-cognate or non-proteogenic amino acids, lot of proteins are shown to be more stable and they are resistant to proteosomal degradation, and because of which we will have prolonged proteins which are active, and these non-cognate or non-proteogenic amino acids are shown to give novel functionalities to the protein, and they are also shown to enhance the function of those proteins. And ultimately, these non-natural building blocks, by sequentially incorporating those in the growing polypeptide or in the growing polymer chain, we can utilize those in therapeutics and bioplastics, and even in the generation of antimicrobial drugs. And ultimately, in our own research, we always struggle to precisely localizing the biomolecule inside the system. By incorporating the fluorescent labeled non-proteogenic amino acid which is having the ability to fluorescence ability, we can precisely get the precise localization of those biomolecules inside the system. But having said that, is it possible, and how it is possible to incorporate these? Because when we are talking about incorporation of these biomolecules, non-proteogenic amino acid, it is not only the translation that we are talking about. We have to start from replication, RNA, we have to start from transcription and translation. So is it really possible to manipulate these machineries of central dogma to incorporate these non-proteogenic or unnatural amino acids? So my talk will be focused on where are we now and what has done and what we are going to see in the near future in the incorporation of non-proteogenic amino acids and also non-amino acids inside through biological machineries. So it all started in 1966, where even recombinant DNA technology was not known, and people have thought, okay, what if we can modify amino acid residue into another proteogenic amino acid residue from a fully folded protein, which is subticillin, where it is having a serine in its active site. They have modified it into cysteine by series of chemical reactions. And after this, we have got recombinant DNA technology, and people have utilized recombination DNA technology to do site-directed mutagenesis in our daily routine. So now, which gave more confidence to the researchers on that time, and they started thinking, if proteogenic amino acids, why not non-proteogenic amino acids? So the first report on incorporation of non-proteogenic amino acids through in vitro methods came in 1989, where people used tRNA ligase to basically ligate the synthetically generated nucleotides, which is covalently bound with a non-cognate amino acid with the already existing tRNA, and they have used in vitro translation system to incorporate this unnatural or non-proteogenic amino acid inside the growing polypeptide chain and ultimately into the protein. So now in vitro is done, then it is in vivo, right? So in 2001, people also showed that it is possible in vivo, that in vitro by charging, by utilizing orthogonal systems. So orthogonal systems are basically enzymes or proteins which can exist in a normal cellular environment, but they cannot have any crosstalk between the host machineries. In this case of orthogonal synthetase, this orthogonal synthetase cannot interact or charge any of the host tRNAs, but it can only charge the amino acid of our interest into the orthogonal tRNA, which will give orthogonal tRNA charged with our amino acid of interest. In the same way, this orthogonal tRNAs cannot be interacting with host synthetases also. So based on which, after this initial report, people have generated multiple amino acid tRNA synthetase and orthogonal tRNA pairs, and because of which charging multiple, which is aminoacylating multiple non-cognate amino acids onto any tRNA was possible. So this is done. Now we have seen chemical mutation, how rDNA technology has helped in this technique, and we have got our in vivo system also. But there was a very huge bottleneck in this system, where if we need to incorporate these non-proteogenic or unnatural amino acids, the ribosomes also should be compatible with it. So people were in need of an orthogonal ribosomes. So if we are talking about orthogonal ribosomes, we should talk about orthogonal mRNAs. So before that, let's see how a typical ribosome recognizes an mRNA. So this is signed Dalgarno sequence and anti-signed Dalgarno sequence, which is present in 16S sRNA. By this recognition, a ribosome recognizes its incoming mRNA. So this both ASD and AD comes in different versions. So these people have mutated these both signed Dalgarno sequence and anti-signed Dalgarno sequences through random mutation, and they have selected and generated orthogonal ribosome and orthogonal mRNA pair. So like any other orthogonal systems, these also can exist in the same cell, but there won't be any crosstalk between normal ribosome and an orthogonal mRNA, and orthogonal ribosome and a normal mRNA. So this has created an entire translation system where we can incorporate non-natural or non-proteogenic amino acids in the protein. But did it stop here? The answer is no. So people again asked the question, if non-proteogenic amino acid, why not non-amino acids? So in 2024, a groundbreaking paper came in Nature, where people have showed that not only non-cognate or non-proteogenic amino acids, but also non-amino acids like beta-hydroxy amino acids can be incorporated in the protein. And they didn't stop there. They have showed that it is possible to incorporate these into the protein, and they purified it and they saw the structure of it, and they have confirmed that it is possible. Now we have seen about orthogonal systems, but is it only specific for translation? The answer is no. People have also worked on orthogonal RNA polymerases and orthogonal DNA polymerases, which means by using these three, we can actually make an orthogonal central dogma. But here also another bottleneck is existing, which is all these are existing in their old niches. They are there in their old systems. It has not been coupled together yet. But a recent report has coupled orthogonal transcription and orthogonal translation system in order to incorporate non-cognate and unnatural amino acids in the protein. So keeping this aside, let's talk about another hand of synthetic biology, which is synthesizing an entire genome, and based on which we can basically generate a bidirectional genetic isolation, which is after generating this bidirectional genetic system, it is not possible for a genetic material to transfer, to be transferred inside the system and outside the system. If that happens, it cannot be translatable. So with this, where are we leading to? Now we have an orthogonal central dogma and we have host central dogma. So by soon we are going to see that these two, which is orthogonal central dogma and host central dogma, can exist in the same system, but they will be mutually exclusive to each other, and they will be working, and I am very sure that these are going to create miracles in both synthetic biology and also in the biology field. Thank you.
Kaushik, that was an absolutely thank you for sharing your insights with us. Now the floor is open for questions.
Kaushik, that GFP that we have is unnatural.
Yes. Yes. So they have they incorporated it in a specific point where it is not going to affect the fluorescence. So like that only they identified that it is fluorescing and they purified it.
And was it stable and was the half-life similar to wild type GFP?
Sorry, sir.
Look at things like aspartic acid or glutamic acid. Not the usual alpha, but you can go to the terminal, which is, for example, delta aspartate. So I had actually made poly delta aspartic acid, poly epsilon likewise.
Yes.
Okay. Now, it's not very clear whether they can be translated or not.
Yes.
But it looks like even they can be part of your what you call the orthogonal thing, okay?
Exactly, sir.
It may be worthwhile having a look at that.
Sure, sir.
That's what I thought, okay?
Sure, sir.
Because as opposed to the host, you're now talking about modifying. Maybe an interesting thing to do.
Yes, sir.
We'll talk about this later.
Yes, sure. Thank you. Yes, sir.
Can you give some examples of where these applications are taking? Where you have synthetic proteins. Give some examples so that enlighten us on that. Like, this is going to be really useful.
So applications wise, so basically like all these proteins, especially with non-proteogenic or unnatural amino acids, they cannot be degradable. So with a normal proteasomal degradation system, and because of which their stability can be increased, especially when we are talking about some of the industrially important proteins, which can be these can be utilized, and their
So then you have to incorporate these, amino acid synthetases or transferases into these organisms,
Yes.
and then engineer
Yes,
them and
yes,
then make.
yes.
But then it need not necessarily, because you also have the cognate amino acids available.
yes.
So they must be competing for this.
No, that's what the beauty of this orthogonal systems. So there can never be a crosstalk between the orthogonal tRNAs and the normal synthetases. So orthogonal tRNAs are very specific for orthogonal synthetases. So there can never be a crosstalk.
Kaushik, in my orthogonal system, say I have to only change one amino acid and rest amino acids would be my proteogenic amino acids. Will my orthogonal system incorporate those into a polypeptide chain?
Yes, yes. So that's how this orthogonal ribosomes, in that case, they can recognize non-proteogenic amino acids also, and they'll work as a normal ribosomes also.
Then it is a
Ribosomes
crosstalk, right? Because it is able to identify the cognate
Yes.
tRNA and tRNA.
So that's there the mRNA plays an important role. Okay. So this, since when I have explained also orthogonal ribosomes with orthogonal mRNAs, right? So they are specific for each other. So without which we cannot form any polypeptide chains, right?
And one more question, how this will affect the cellular homeostasis, because there is an altogether a separate system which is very stable and not degradable?
Yes, so initial work on this has shown that it is completely lethal to the cellular system, but now after advances they have showed that it is not lethal. They made it orthogonal systems by incorporating a lot of advances, they made it non-lethal. Yes.
We'll take one last question.
Did they change anything at DNA level to incorporate this non-proteogenic amino acid? Like how the same codon can incorporate a different non-proteogenic amino acid rather than a cognate amino acid?
So that's how one of the limitation was. So they have changed. So they have refactored the one codon for another amino acid, proteogenic amino acid. They have refactored it. That's there. But the whole central dogma is not been coupled yet. So that's what I feel the advances will be. But yes, indeed, like they have refactored for instead of one proteogenic amino acid, they have made the system to incorporate another proteogenic amino acid. Example, like instead of serine, they made alanine. They made alanine to be incorporated, or histidine, or proline, leucine. Yes, but not in the case of non-proteogenic amino acid yet.
Thank you so much for that, Kaushik.